Welcome to the Deep Dive, where we take complex information and distill it into clear, actionable insights just for you. Today, we're unraveling something many of us encounter but maybe don't fully grasp: the metabolism of ethanol.
Alcohol. Yeah, I was thinking about our listener, Ivan A., who mentioned hearing that calories from alcohol don't count, like they're just empty calories. He was wondering if they really don't cause weight gain. That's a common idea. But it does make you think, what actually happens when we drink? Where does that energy go? What are the real effects inside our bodies?
That's a great question to kick things off. And our mission today is essentially to, well, break down exactly how your body processes alcohol. We'll trace its biochemical journey, from that first sip all the way to its broader impact on health. We might uncover some surprising things too. Think of it as your guide to what's really going on metabolically.
Okay, let's unpack this then, right from the start. You have a drink.
hmm
Where does the ethanol actually go? How fast does it all happen? Right. Well, ethanol is pretty unique. It dissolves in both fat and water. That means it gets absorbed incredibly quickly from your intestines straight into the bloodstream. A very small amount, maybe 0 to 5%, it gets metabolized right there in the upper GI tract. But the vast majority, we're talking 85 to 98%, heads straight for the liver. That's the main processing plant. And only a tiny fraction, maybe 2 to 10%, just leaves the body through breath or urine. So once it gets to the liver, the main pathway kicks in. It's basically a two-step oxidation process. First step. Ethanol is converted into something called acetaldehyde. It's not a pleasant compound. This is done by an enzyme, alcohol dehydrogenase, or ADH. And crucially, during this step, a molecule called NAD+ gets converted into NADH. Think of ADH as taking hydrogens off the ethanol and giving them to NAD plus LA. This change in the NADH to NAD+ ratio, it's going to be really important for understanding the immediate effects. Then step 2. That acetaldehyde is pretty toxic stuff, so the body wants to get rid of it fast. It gets oxidized again, this time into acetate, which is relatively harmless. This happens using another enzyme, acetaldehyde dehydrogenase, or ALDH. And guess what? This step makes more NADH.
Okay, so the liver's suddenly flooded with NADH. Got it. And this acetate, you said it's non-toxic. What happens to it? Does the body just ditch it or use it?
Good question. Most of that acetate actually gets released back into the bloodstream. And other tissues, especially muscles, love it as an energy source. They grab the acetate, convert it into acetyl-CoA, and feed it right into the TCA cycle, the main cellular powerhouse to generate ATP, energy. The liver can use acetate itself, maybe to make fats or cholesterol, but typically it prioritizes shipping it out for fuel elsewhere.
Okay, that sounds pretty straightforward for the main route. But you hinted, this is where it gets more interesting. Are there other ways the body handles ethanol, like backup systems?
Absolutely. There's a secondary route called the microsomal ethanol-oxidizing system, MEOS for short. Now, this pathway usually only handles maybe 10 to 20% of the ethanol in someone who drinks moderately. It also turns ethanol into that toxic acetaldehyde, but it uses different machinery. Specifically, cytochrome P450 enzymes, particularly one called CYP2E1, located in a part of the cell called the endoplasmic reticulum. And unlike the main ADH pathway, this MEOS system actually uses up energy in the form of NADPH, and it needs oxygen. What's really key about MEOS, though, is that its activity increases quite a bit when there are high levels of alcohol or with chronic drinking. That's because the body actually makes more of the CYP2E1 enzyme. It's inducible, and it handles higher concentrations better than ADH.
That makes sense. And that leads to something you often hear about how differently people react to alcohol. If these enzyme systems vary, does that explain why?
Exactly. There are genetic variations, polymorphisms in all these key enzymes, ADH, ALDH, and even CYP2E1. These genetic differences are a major reason why people clear alcohol at different speeds, feel its effects more or less intensely, and why some are more susceptible to damage. There's a well-known variant of the ALDH enzyme, ALDH242. It's much slower at breaking down acetaldehyde. So people with this variant get a rapid buildup of toxic acetaldehyde when they drink, causing things like facial flushing, nausea, vomiting, really unpleasant effects.
Wow, so that unpleasant reaction actually protects them.
In a way, yes. It often makes drinking so uncomfortable that it strongly discourages heavy consumption and lowers the risk of developing alcoholism.
Okay, so we've got these pathways and this big shift in the NADH-NAD+ ratio you mentioned. Let's focus on that. What are the immediate sort of metabolic consequences of that NADH buildup?
Right, that NADH surge is central to many of the acute effects. Think of it as gumming up the works in the liver's metabolic machinery. It really throws things off balance. So, first off, high NADH levels put the brakes on fatty acid oxidation. The liver just can't burn fat effectively for energy. Instead, those fatty acids pile up and get repackaged into triglycerides. This leads, even after just a moderate amount of drinking, to fat accumulation in the liver, what we call a hepatic steatosis, or fatty liver. Chronic drinking makes this much worse, partly because alcohol also stimulates pathways that make fat.
Okay, so fatty liver is one immediate thing. What else?
Well, that NADH imbalance can also trigger something called alcohol-induced ketoacidosis. The high NADH essentially diverts key molecules away from the normal energy cycles. Acetyl-CoA gets sort of trapped and can't enter the TCA cycle easily. So, the liver compensates by converting that acetyl-CoA into ketone bodies. If this goes too far, your blood can become acidic. Ketoacidosis.
Is that like the clinical example you mentioned, LM, the semi-conscious patient?
Precisely. His symptoms, the rapid breathing, the lab results showing a high anion gap, low blood sugar, but high levels of ketone bodies like beta-hydroxybutyrate pointed directly to this. The alcohol metabolism, probably combined with not eating, pushed his body into making excessive ketones.
That sounds serious. Are there other acid-base issues?
Yes, there's also lactic acidosis. Again, the high NADH pushes the conversion of pyruvate towards lactate. So lactate builds up in the blood, contributing to acidity. And as a side effect, high lactate can actually interfere with the kidneys getting rid of uric acid, which can be bad news for people prone to gout.
Okay, and what about blood sugar? I've heard alcohol can mess with that too.
It definitely can, and it depends on the situation. In someone who's fasting hasn't eaten, that high NADH strongly inhibits gluconeogenesis. That's the liver's process for making new glucose. Key precursors get diverted away. So you can end up with dangerously low blood sugar hypoglycemia. This is a serious risk, especially with binge drinking on an empty stomach.
But sometimes it goes the other way.
It can, yeah. If you drink with a meal, ethanol can actually interfere with how your cells use glucose, potentially causing a temporary spike in blood sugar or hyperglycemia by inhibiting glycolysis.
It's really striking how much just a few drinks can derail normal metabolism. But let's shift gears now. What happens when those occasional drinks become a regular chronic habit? That's when we start talking serious long-term liver damage, right?
Absolutely. The chronic toxicity is largely driven by the persistent effects of those two main culprits we identified, acetaldehyde and free radicals. Acetaldehyde, remember, is highly reactive. Over time, it continuously forms these damaging adducts with proteins, DNA, lipids, basically essential cellular components.
And what do these adducts do?
They cause a lot of problems. They impair the liver's ability to synthesize crucial proteins, like albumin for fluid balance, and clotting factors needed for blood coagulation. They also mess with the secretion of VLDLs, the particles the liver uses to export fat, so fat keeps accumulating. And as these damaged proteins and fats build up inside liver cells, they draw water in, causing the cells to swell. This disrupts the liver's delicate architecture and can contribute to increased pressure in the liver's blood supply portal hypertension. Plus, acetaldehyde weakens the liver's own antioxidant defenses, like glutathione, making it even more vulnerable.
A vicious cycle then. And you mentioned free radicals too. Where do they come in?
They're a major part of the chronic damage, especially linked to that MEOS pathway and the CYP2E1 enzyme. When CYP2E1 metabolizes ethanol, it generates a significant amount of reactive oxygen species, ROS, free radicals. Think of them as unstable molecules that attack other cell components. They particularly damage cell membranes, including membranes of the mitochondria, the cell's power plants. This process is called lipid peroxidation. Damaged mitochondria can't produce energy efficiently, and they become leaky. This further impairs the oxidation of acetaldehyde, worsening the toxicity. It just spirals.
So all this damage, the acetaldehyde adducts, the free radicals, the mitochondrial dysfunction, how does it manifest as actual liver disease?
It leads to the progression we call alcohol-induced liver disease. It typically moves through stages. Stage 1 is that hepatic steatosis, the fatty liver. At this point, it's usually reversible if the person stops drinking. Stage 2 is alcohol-induced hepatitis. Now you have significant inflammation, liver cell injury, necrosis, and cell death. This might be reversible, but it's much more serious. And stage 3 is cirrhosis. This is the end stage, characterized by extensive, irreversible scarring fibrosis. The scar tissue replaces functional liver cells, completely disrupting the liver structure and function. This ultimately leads to liver failure.
Like in the case of Jen T you mentioned, the artist with fatigue and jaundice.
Exactly. Her symptoms, the fatigue, the liver pain, the jaundice, and her lab results showing elevated liver enzymes like ALT and AST, plus high bilirubin. These all pointed towards significant liver inflammation and impaired function, consistent with ongoing hepatitis and the development of cirrhosis due to her chronic heavy drinking.
You also mentioned the acetalt ratio being useful.
Yes. In alcoholic liver disease, typically the AST level is higher than the ALT level, often by a ratio of 2.1 or more. In many other types of liver injury, like viral hepatitis, the ALT is usually higher. It's a helpful diagnostic clue, but not definitive on its own. And that scarring process in cirrhosis, it involves specific cells. Damage signals activate immune cells called Kupffer cells. These then release inflammatory signals and growth factors, like TGF-beta1, which in turn activated another cell type, the stellate cells. Normally, stellate cells are quiet, storing vitamin A. But when activated, they transform into myofibroblasts, basically scar-producing factories. They churn out huge amounts of extracellular matrix proteins, especially collagen, leading to that dense fibrous scarring that chokes the liver.
And you mentioned something about gene regulation, too, SART1.
Right, that's another piece of the puzzle. Chronic alcohol seems to decrease the activity of a protein called SIRT1. SIRT1 normally helps regulate metabolism, partly by activating AMPK, which usually promotes fat burning and inhibits fat synthesis. So, when alcohol suppresses SIRT1, AMPK activity goes down, and this contributes to increased fatty acid and triglyceride synthesis in the liver, worsening the fatty liver situation.
So once cirrhosis sets in and the liver structure is destroyed, what are the really critical functions that fail?
It's a systemic failure, really. The liver can't make enough albumin, leading to fluid imbalances like swelling, edema, and fluid in the abdomen, ascites. It can't make clotting factors, so bleeding becomes a major risk. Toxic substances build up. Ammonia, normally converted to urea by the liver, accumulates in the blood and can affect the brain, causing hepatic encephalopathy. And bilirubin builds up, causing that deep jaundice we saw in Gene T. Essentially, the body loses its central metabolic and detoxification hub.
Okay, let's circle back to Ivan A's original thought about empty calories. Now that we've dissected the pathways, can we give him a clearer answer on the energy yield? Why might heavy drinking not lead to weight gain?
Yes, let's tackle that. It's nuanced. As we said, the main ABADH pathway is pretty efficient, generating about 13 ATP per ethanol molecule, so those 7 kilocalories per gram of alcohol. For moderate drinkers, they definitely contribute energy.
So Ivan wasn't entirely wrong initially.
For low levels, no. Those calories count towards your total energy intake. But when you get into chronic heavy drinking, the MEOS pathway plays a bigger role. And remember, MEOS consumes energy in ADPH, so its net ATP yield is lower, maybe around 8 ATP per ethanol. On top of that, you have the mitochondrial damage we discussed. Oxidative phosphorylation gets uncoupled. This means the energy released from breaking down fuel isn't efficiently captured as ATP. Instead, more of it is lost as heat.
So less usable energy is actually generated and some is just wasted as heat.
Exactly. So despite consuming a lot of calories from alcohol, the body might not be capturing and storing that energy as efficiently as it would from, say, carbohydrates or fats. That helps explain why some chronic heavy drinkers don't gain as much weight as you'd expect based purely on caloric intake. So the calories aren't truly empty of energy, but they're less efficiently utilized, especially over time, and they definitely lack essential nutrients.
And that increased MEOS activity, the CYP2E1 induction, that relates to tolerance too, right?
It does. Producing more CYP2E1 means the body can clear ethanol from the blood faster. This is a major part of metabolic tolerance. The chronic drinker needs more alcohol to achieve the same effect because their body breaks it down quicker. But, and this is crucial, faster clearance via MEOS also means faster production of toxic acetaldehyde and more free radicals, increasing the risk of liver damage. Tolerance comes at a steep price.
It's a complex picture. And we shouldn't forget nutrition's role in all this, should we?
Absolutely not. Alcohol directly interferes with how your gut absorbs key nutrients, things like folate, thiamine, vitamin B1, vitamin A. It also messes with how the liver stores and activates vitamins. So chronic drinkers often become severely malnourished, even if they're consuming enough calories. This malnutrition drastically worsens the liver damage and overall health outcomes.
And briefly, you mentioned drug interactions earlier.
Yes, it's important. Because ethanol interacts with those cytochrome P450 enzymes, it can really affect how other drugs are metabolized. Depending on the specific drug and the pattern of alcohol use, it can either speed up drug breakdown, making the drug less effective, or slow it down, leading to toxic drug levels. Mixing alcohol and medications, especially things like sedatives or acetaminophen, can be very dangerous.
Okay, we have covered a huge amount of ground here. Let's try and summarize the key takeaways for everyone listening. First, ethanol's journey mainly happens in the liver via two key routes, the ADH-LDH system and the MEOS system. Both end up converting ethanol to toxic acetaldehyde and then finally to acetate.
Right. Second takeaway. Those immediate acute effects, largely down to that big increase in the NADH and AD plus ratio in your liver. This metabolic imbalance causes things like fatty liver, ketoacidosis, lactic acidosis, and potentially dangerous hypoglycemia if you haven't eaten.
Third, the really serious long-term damage comes from the chronic toxicity of acetaldehyde and free radicals generated during metabolism. This damage progresses from that initial fatty liver, potentially through inflammation and hepatitis, to the irreversible scarring of cirrhosis and liver failure.
And fourth, how alcohol affects you specifically depends heavily on individual factors. Your genetics play a huge role in those enzyme variations. Your drinking history influences tolerance and MEOS activity, gender matters, and of course, how much you drink is critical.
And finally, for Ivan A. and anyone wondering about those empty calories, alcohol does provide energy, about 7 kilocalories per gram. But in chronic heavy drinkers, the metabolic processing becomes less efficient, especially via the MEOS pathway, and energy can be lost as heat. Plus, those calories completely lack essential nutrients.
Exactly. Understanding these sort of biochemical details really highlights the incredible complexity inside us. It shows why responses to alcohol can be so incredibly varied between different people. It definitely raises some important thoughts about personal health choices, maybe public health approaches too, and just how much our individual biology shapes our experience.
That's a great place to leave it. Thank you so much for walking us through all of that, and thank you for joining us on this deep dive into ethanol metabolism. We hope it's given you a clearer picture. Keep learning, keep questioning, and we'll catch you on the next deep dive.